Okay, hello everyone. Welcome to this session of webinar on tuberculosis, the silent killer, understanding disease dynamics, diagnostics and treatment. I'm Rakesh Mishra, and it's my pleasure to welcome all of you for this seminar. We have very accomplished speakers, four very accomplished speakers. Dr. Vijay Nandikuri, Director of Center for Cellular and Molecular Biology, Hyderabad. Dr. Vadrajan Sundarmurthy, a Senior Scientist, Faculty at National Center for Biological Sciences in Bangalore. And Dr. Sadhna Sharma, Professor and Head of the Biochemistry Postgraduate Institute of Medical Education and Research in Chandigarh. And Dr. Urvashi Singh, Professor and Chief of Tuberculosis Section, Department of Microbiology, All India Institute of Medical Sciences, New Delhi. So we have a really collection of speakers who are very accomplished and represent premier institutions of various specialty in the country. And once we have heard all the speakers, we will have a brief message of about 5 minutes from Dr. Soumya Swaminathan, who has not been able to join, but gave the message. She herself as a major personality in medical field in the world, and also a specialist of tuberculosis as a career. So before I introduce the speakers, it's my privilege to introduce to you a little bit about what we do at Tata Institute for Genetics and Society, and why we are interested in seminars of this kind by inviting the prominent speakers and spreading the message of where things stand in major health problems in the country. So TICS, as we call it, is our institution located in NCBS INSTEM campus in Bangalore, and is a non-profit research institution whose interest is to use cutting-edge technology to solve societal problems for which problem exists, technology exists, so we can connect and provide the solution to the society. And we have three major research programs in TICS. One is about crop improvement, where we use the genome editing technologies and introduce quality or traits in the plants which are required now for nutrition and environmental reasons, because we don't have resources to grow the crops the way we are able to do today. Then we have another major program in rare genetic diseases, where we address both diagnostics and interventions in rare genetic diseases, which are ignored because of the number and because they are unique to our country, where multinationals don't come in, and wherever there are solutions available, they are prohibitively expensive. They cause in crores, so that's not possible. So we try to bring those things in the reach of our society. And the third program that we have, which we are linking this webinar to, is infectious diseases, in which we do surveillance and disease ecology. Is one component. We look for vector control that spread the disease, particularly mosquitoes, and also work one vertical on antimicrobial resistance, which is probably going to be a bigger problem than even current pandemic in the near future, unless we attend to it. And we have a specialist speaker, which I hope will cover some aspect of those in their talks. So we are in tuberculosis is something which the moment you say infectious disease, this is probably the first name that comes, and it has become even more important when we have any immune situation where immunity is compromised. Then this silent disease becomes more aggressive and leads to very poor health conditions. And so government of India has plans to get rid of tuberculosis completely. Lots of efforts are going on, and it's a right time to discuss about this and what are the progress and where we need to put more resources into. TICS will be particularly interested in looking at if there are AMR components where we can make some, bring some interventions or bring in such diagnostics at a point of care diagnostic where we can clearly identify, estimate very quickly, and then take interventions, take the interventions there so that the disease can be eliminated as our government is hoping for. So, and I think we will, after these 4 talks that you are going to have in next 1.5 hours, we'll all be more informed, more up to date with this field. And in the meantime, during the presentation, any one of you have questions, please go to chat box and type your questions. And our experts will, the speakers, whenever they have time, some of them they can answer directly there, or after the talks, we will bring some of those questions and try to answer you. So with that, we should now start because so that we finish in time. So I would like to really introduce first speaker, Dr. Vinay Nandikuri. It's my pleasure to introduce Dr. Nandikuri. He's a renowned biotechnologist and currently the director of Center for Cellular and Molecular Biology in Hyderabad. He is well known for his studies on the kinase-mediated signaling network in Mycobacterium tuberculosis, the causative pathogen of tuberculosis. He holds a master's degree in biotechnology from the Indian Institute of Technology, Bombay, and a PhD from the Indian Institute of Science, Bangalore. Dr. Nandikuri did his postdoctoral work in the University of Virginia in Texas. He is a recipient of National Bioscience Award for Career Development, one of the highest Indian Science Award, and his contributions to biosciences from the Department of Biotechnology, Government of India. He is a Narcissus Cooper's Young Scientist Award. He is also elected Fellow of the National Academy of Sciences in India. With that, I'll request Vinay to start his presentation.
Thanks, Rakesh. I was waiting for it to get over. Thank you. That was long. Can you see the screen?
Yes.
Okay.
Yeah. Good.
Since I'm the first one to go, I guess I will have to give a bit of introduction about tuberculosis. I am going to talk to you about pathogen aspects. I'm not going to talk to you about the host. That's where I think Varda will take over. I currently have 2 labs, one at National Institute of Immunology, where I have 4 members currently, and the other one I started at CCMB when I moved last year. I have around 7 or 8 members there, and this is a photograph of everybody who's currently working in my lab. As Rakesh mentioned just a while ago, when it comes to infectious disease, it is not the SARS-CoV-2 which is the biggest killer. Unfortunately, it is still tuberculosis which is the biggest killer. That's because SARS-CoV-2 is a recent infection. It has killed approximately 30 lakh people every year for the last 2 years. But tuberculosis has been around for centuries and actually literally thousands of years, thereby killing a lot more people over a period of all these years than any other infectious disease there is known of. How do you get tuberculosis? It's aerosolic in nature. You can encounter a person who has tuberculosis and those droplets enter your body. They are immediately taken up by macrophages. But tuberculosis has a knack to survive inside those macrophages. And I did not say the organism's name is Mycobacterium tuberculosis. So once it is there, the host goes to the other end, which is basically gets in all the other immune cells to contain the infection. And it forms something called a granuloma when all the adaptive immune response comes into play. Under these circumstances, what you have is a granuloma is has 3 different fates. If your immune response is extremely good, you would completely eradicate it. But if it is not so good, you might actually have a very active disease, and then it spreads to other parts of the lung and to the extrapulmonary parts of your body, right? The other one is the latent tuberculosis, which is what happens in majority of the cases, where your tuberculosis is still there in your body, but it is not an active disease. When the situation is appropriate, such as people having an HIV infection in parallel, or some other disease, somewhere tuberculosis gets activated, then you will end up having a full-blown disease at a later point of time. I've been investigating the various concepts, which include signaling. Conceptually, whatever you want to talk about in the context of any cell, whenever there is an extracellular stimuli, your cells are expected to respond, and that is simply what signaling is. Signaling can be in many forms. It can be transcriptional response. It can be many other responses. We have been looking at delineating molecular mechanisms that happen in Mycobacterium tuberculosis for the last 18 years. My lab works on multiple aspects, which is one of the things which Rakesh mentioned about is serine, protein kinases, phosphatases. We have a strong body of work in this. And we've also worked on peptidoglycan synthesis, cell division, host-directed therapy, even including evolution of drug resistance. Last time I presented evolution of drug resistance in NCBS, so I'm steering away from that at this point of time. So I would like to today talk about our regulation of transcription work. One of that is published in EMBOJ last year, but I'm going to talk about something very different. There are approximately 200 transcription factors in this pathogen. And out of these 200 transcription factors, 13 of them are sigma factors. So this work, before I go to the work, I will tell you about the people who have worked. Biplab is the person who has actually done all the work that I'm going to present today, supported by Mehak, and this was done in collaboration with Gopal and Debashri from ISC Bangalore. Divyatej and Nitish Kumar from CCMB have also helped in this project. So coming back to the sigma factor, there are 13 sigma factors present in Mycobacterium tuberculosis. And any sigma factor's basic function is to recruit RNA polymerase so that the transcription can take place in your body, whether it is a E. coli or us. I mean, transcription is a basic function. So there are four groups of sigma factors. Those sigma factors which only contain minimal domains required, one recognizing -10, one recognizing -35, is called group 4. And if you have more than that, which also gives you a higher level of specificity, extended -10 with a domain 3, that's group 3. And you have one more which also looks into discriminator region between -10 and transcriptional start site, that's group 2. So there is only one group 2 and one group 1 to sigma factors, one in case of Mycobacterium tuberculosis. Sigma factor, group 1 sigma factor has everything plus an N-terminal extension. So many of the sigma factors are kept in the cellular membrane, and when the need arises, they are released and they're kept in check with an anti-sigma factor. When the need arises, the anti-sigma factor gets degraded and they become free and actually respond to the stress that there is. So they respond to variant stress as indicated, DNA damage, oxidative stress, and various other things. But anyway, for today, I would like to focus on the primary sigma factor because nobody realistically has done any biological work on this before we started out. So here is a crystal structure of sigma factor in complex with RNA polymerase, RBP, and all those things, right? And DNA. So based on this is a structure we know quite a lot of interactions sigma factor makes with the DNA and with the other proteins. It is the housekeeping sigma factor. You take it out and cell supposed to die because there is a high throughput mutagenesis that showed it. It is essential for the survival of the pathogen, and it's a complex structure is available. However, there is no study whatsoever on the in vivo functionality of the sigma A or how important these interactions are. So we set out to do this around 5 years ago. Basically, for doing these kind of things, you need to generate a conditional mutant. How do you generate a conditional mutant? You put an inducible copy elsewhere in the genome, and then using homologous recombination, knock out the native loci. That's precisely what we have done. We put an additional copy, which is TET regulated, which in the absence of tetracycline, actually expresses the RNA and hence the protein. But in the presence of tetracycline, repressor binds to the operator sequence, shutting down the expression of the gene which is under a particular regulation. So it's a TET off system. What we did is we put an additional copy first in the Mycobacterium tuberculosis genome and looked at its regulatability by providing anhydrous tetracycline. You can very clearly see it is going down here. And then we took out the native loci using an phage-mediated transduction and confirmed that with multiple PCRs. I really don't want to go into the details, but we have confirmed with multiple PCRs and followed it up with a Western blot confirmation. Very clearly, you can see native loci copy is without a tag, so it is smaller. The one with the flat tag is runs slower, and when you add ATC, it is gone. So how do you know how do you actually evaluate the function of any of these things? One does it at in vitro, which is simply growth in culture. Second thing is a cell line experiment. The third would be a murine infection experiment, and murine infection experiment in an established infection. We have done it all. I'll go stepwise. To start with, in vitro, what we have done is the first thing is we checked out what happens to the mutant when you add anhydrotetracycline to the media. So what you see is without anhydrotetracycline, what you see is this. And in the presence of anhydrotetracycline, it is added on day one. You can see a death happening as we are going towards different days of growth. So the same strain without anhydrotetracycline, same strain with anhydrotetracycline, you can very clearly see a death. The same experiment we have performed in peritoneal macrophages isolated from the mice, and the infection was carried out for 96 hours using these strains, either the RV or the RV delta Sig strain, in the absence or in the presence of ATC. You can very clearly see a death happens when you actually deplete sigma A. These things are kind of giving us the same thing that we kind of know, but nevertheless, one has to prove it multiple ways. So we have done next is a mice infection experiment. In this experiment, what you do is you challenge the mice aerosolically with various strains. And after 30 days, you basically look at the bacterial load in the lungs and the spleen. What we got is that in the wild type, and how do you know this? In this case, we provide doxycycline in the water for the mice. When you provide doxycycline, you get hardly any load, and most of the bacteria is eliminated in these mice. And here is what you get without anhydrotetracycline, and here's an RV with anhydrotetracycline, nothing because happens. Same thing was observed even in the spleen. But nobody comes to a hospital anticipating an infection. They come to your hospital after having an infection. So is sigma A a good drug target? To check this out, what we have done is we allowed the infection to be established for a period of 2 weeks. After 2 weeks, we started giving, put it into groups and started giving doxycycline to them. This is a wrong figure, but only focus on the 6 weeks and 10 weeks point of view. You can very clearly see that even after establishing the infection for 2 weeks, when you give it doxycycline, you can very clearly see a downregulation. And that continues to be the same, but it doesn't die any further, which may be because other sigma factors take over in the chronic phase of infection. So all these things tell us that sigma factor is essential and sigma factor can be a good drug target. So, but sigma factor interacts with many things, including the promoters. How important are these interactions with respect to the promoter? So since we have a crystal structure, we knew the residues that are involved in these interactions. So for example, there is a -10 interactions is through these tryptophan residues, and this threonine and arginine residues contribute to -35 interactions. So what we did is we did complementation experiment. Means you put it an additional copy, episomal copy, and then you add anhydrotetracycline and see if the episomal copy complements. So RV without or with ATC gives the same. Delta Sig without or with ATC gives you death, addition of anhydrotetracycline. If you provide an additional copy of sigma A, you get a complete rescue. But if you provide a sigma A which does not have -10 interacting residue, it's almost as good as the vector alone. And when you provide something without -35, however, there is a partial recovery. It's not, there is death, but it's not comparable to without sigma A. Then we looked into what happens with sigma A interactions with other proteins that are known to be interacting. And again, we know the residues. So we mutated those residues in sigma A and complemented with the sigma A, which has mutations in those residues. What did you see here also is that you take out RBPA or you take out YB interacting residue, we get death, but not as much as we get without having sigma A altogether, which I call it partial, but nevertheless it is death. So in addition to all these things, since my long-standing interest in life is all about phosphorylation, we looked into the literature and found that sigma A is phosphorylated on 3 residues in vivo. So we basically made phosphoablative, where you convert them into either alanine residues or mimetic, where you put it into either a D or a E, aspartic acid as glutamic acid, and then you perform the rescue experiments. What we got is that when we converted all three amino acids into an alanine residue, we ended up losing some level of death happens. When we convert them into phosphorylated mimics, we do get some death. So it appears that the phosphorylation is some level of regulatory, and the hydroxyl residues that are present in this S and T play a role in modulating the sigma A functionality. So what is the, these are all good, it's a transcription factor. What is the impact on the global transcription? It was very easy. We could not deplete too long because it was dying. Too less, we were not getting any phenotype. So we depleted for 3 days, and we looked into what happens. What is interesting is approximately 57% of the whole MTB transcriptome changed when you don't have sigma A. Kind of not very surprising because it's a housekeeping sigma factor. Change was very drastic, you can very clearly see. And around 1098 genes were downregulated, which you can call are the direct targets of sigma A. And there are 1168 genes are upregulated, which may be because of the indirect phenomena, like, some of the repressors now are downregulated, and hence the transcription may be upregulated for the other things. Whichever way, sigma A seems to drastically change the transcriptome whole profile. So we basically started looking at these results, and we find that whenever sigma A is downregulated, sigma B, which is another sigma factor, was upregulated. And this was consistently observed both in QRT PCRs that were done to validate the study, as well as in Western blot. So we started wondering, is there some kind of a parallel between sigma A and sigma B? The difference is only in the N-terminal region. Can overexpressing sigma B complement the functionality of sigma A? So this was a very obvious question that we asked. So what we did is we actually took out RV delta sigma B also we created, but we put in either sigma A or sigma B to complement. But though it appeared that way, overexpressing sigma B in the sigma A background knockout did not actually give us an E-rescue. So while there seems to be an indirect response, it doesn't seem to have a major role. But then what is the role of sigma B and sigma A? What is the overlap? We already had a knockout, so we simply went ahead and performed what we RNA-seq experiment. So what we have found is 438 genes were regulated differentially, and that is around 1/5 of what is happening with sigma A, and out of which hardly any of them overlap. 2000 genes are completely solely modulated by sigma A alone, 150 by sigma B, and around 160 by sigma B, and these are the ones which are modulated by together. And there are some explanations, but it's interesting enough that they seem to have a completely independent set of functions. So that led to the last question. What is the difference? The difference is in the N-terminal region. So what we did is any molecular biologist will do. We systematically deleted the amino acids from the N-terminal and started looking into what happens. Since we were also collaborating with BG, we have done both biophysical studies and in vitro transcription work. In biophysical studies, all these fragments were interacting with DNA with very similar efficiency. So there is no problem of interacting with the DNA. But in vitro transcription, when we performed, the delta 179 sigma A could not give you any in vitro transcription. So it binds, but it does not lead to transcription. Maybe it is not converting from a closed complex to an open complex. In vivo, these results were mirrored. All of them rescue other than the delta 179. So what is there between 132 and 179? We observed that there are approximately a stretch of acidic amino acids, and these acidic amino acids are basically most probably required for converting from an closed complex to an open complex during the transcription. So this is a speculation at this point of time. So, this is still an ongoing work. And I thought anyway, I'll present something which is more ongoing so that I might get suggestion. Sigma A is essential in vitro conclusions. Whatever we have done is this ex vivo, in vivo. Sigma A interaction with the promoter element and other proteins plays a significant role. Sigma A regulates 57% of the transcriptome, has unique non-redundant role, and the N-terminal domain and phosphorylation are critical. Where are we going with this? So I am almost taken up my time. I really don't want to take any longer than that. But where are we going with this? Now, what we have realized is that there seems to be subsets that are controlled by different interactions of sigma A. We would like to next go ahead and find out what are the promoters that are bound by sigma A by performing ChIP-seq. And we would like to see, and if there is any variation, if you abrogate RBPA interaction, if you abrogate YB interaction, and if you abrogate -35, or if you have phosphorylation site mutants. So this is something that we will be taking forward. But at this point of time, where are we is basically where are we is that we basically have done this. And also something that I can completely say is unpublished is that we have done an interactome and found some very, very novel interacting partners. Now we are working on what is this novel interacting partners are doing in the context of sigma A. So transcriptional regulation is complex. It requires a lot more intricate study from our end. This is where we are at this point of time. And that's where I would like to stop. Thank you very much.
Thank you. Thank you very much, Vinay, for a very simplified and very nice discussion of this pathogen. We will have question answers once all the speakers have finished talking. So I request all participants to type their questions in Q&A part, and we will keep them getting answered as and when the speakers get time. Otherwise, we'll address them later after the talks are over. So we now go to our next speaker. Our next speaker is Professor Varadarajan Sundaramurthy. Dr. Sundaramurthy is a PhD from Department of Biochemistry, Indian Institute of Science, Bangalore. He worked on heme metabolism in Plasmodium. Later, he did his postdoc from the Biozentrum University of Basel in Switzerland on signaling pathways during MTB infections, followed by another postdoc from the Max Planck Institute of Molecular and Cell Biology and Genetics in Dresden, Germany, on high content screening and drug discovery for intracellular bacterial infections. He started his independent laboratory at NCBS in 2014. His current research focuses on understanding the fundamental mechanisms during intracellular infections and with Mycobacterium tuberculosis. And also he has interest in Plasmodium, liver stage, and SARS-CoV-2 with implications for the host-directed therapeutics, antimicrobial resistance, and drug discovery. So, Dr. Varadarajan, please, you can start your talk. Thank you.
Sorry. Thanks, Rakesh, for the kind introduction. And good afternoon, everybody. I'll talk today about some of the work we are doing in the lab on mycobacterial infections. Vinay has given a fantastic introduction, so I'm not going to go into the disease per se. Before I start, I really want to thank all my lab members and the funding agencies and the excellent collaborators and the facilities we have in NCBS. So what we are working on, our lab is looking at the host side of the host pathogen interactions, and Vinay spoke nicely about specific bacterial processes and detailed description of molecules. We are going to at the cellular pathogen host site schematic, where we can see the disease progression of TB, starting from a condition where a non-infected versus a full-blown disease. And in reality, the disease is a spectrum between this entire scheme of possibilities. In a large number of cases, you have the disease, you have the bacteria infecting a person, but for all practical purposes, these people are not showing signs of infection. So they are disease is held in some balance, and like shown in this movie here, there is active forces from both the bacteria and the pathogen which is holding the system in check. So what we are trying to understand in the lab is, broadly speaking, at a cellular level, what are the determinants of this bacterial survival inside the cell? And survival also I include broadly to mean uptake and survival. So here is a picture of cells which are infected with mycobacteria. And this graph here shows a certain quantification of this, essentially how much bacteria is there in the cell. You can see it follows a certain distribution, but essentially what is it that determines that some cells get infected, some cells don't, and in the cells that get infected, why is it that the infection settles to this level, not higher or not lower? So that's the kind of broad contours that we are asking in the lab with a variety of different broader themes. And a lot of questions that we ask in the lab centers on the trafficking milieu of the host. And this is very important because we know for a long time that mycobacteria is modulating the maturation of the phagosomes. I will not go into the details. This will be very familiar to the audience here. But what is really emerging is that the trafficking milieu of the MTB infected macrophage is very complex. We know that the mycobacteria is sitting inside this arrested phagosome, but despite its localization here, it can and does influence the trafficking systems way beyond the confines of the phagosome and also into the nearby infected cells. So in this complex trafficking milieu, what are the kind of interactions that are happening between the bacteria and the host, and how does that influence the bacterial survival? Now, this is a beautiful picture movie that came from David Russell's lab, where they surface labeled mycobacteria and infected the cell. And here you can see the label from the mycobacterial surface is getting out of the bacteria here, and it is actually going all over the cell. You can see this going in discrete puncta all over the cells. And they show that these puncta are essentially concentrating in the endolysosomal system of the cell. So despite the mycobacteria arresting the phagosome maturation, it is having a strong influence on the cell. So what are these influences? So to look at this, we systematically quantified the endolysosomal system of the infected cell. And for the purpose of today's talk, I will talk about the comparison between a mycobacteria infected cell and what we call as bystander cells, which are non-infected cells in the same milieu. And we can see that the uptake of an endocytic cargo is very high, particularly in the infected cells. These are in culture, and this is from cells taken from infected animals. And again, you can see that in vivo also, the infection, the uptake of different cargo like dextrin is very high. And similarly, the lysosomes are very high in infected cells compared to uninfected cells. As early as 2 hours after infection, this is sustained a lot over time. And also in vivo, when we isolate macrophages from infected cells, you see that there is a high lysosome numbers in the infected cells. And we can see that the lysosomal influence is way higher than the endosomal influence. And I will not go into the details, but happy to discuss this a lot. If you look at the difference between the red and the black curves here, essentially the red is staying inside the black. The black is the total population of the cells. So the endosomes are modulated, but they are modulated to a much lower level compared to lysosomes, which you can see here for mycobacteria infection is way beyond the normal limits of the system. And in fact, this is something we can actually predict simply by looking at the lysosomal features of the system. We can actually predict if a cell is infected or not. And we also see this in a context of an in vivo infection, where if we follow the infection over time, we can see that simply there is a very high lysosomal content in these are tissue sections 12 weeks after infection compared to 2 weeks after infection. Not only that, we see that mutants, so the high amount of lysosome in the cell also equates to higher lysosomal delivery of mycobacteria. So despite the arrest of the phagosome maturation, with increase in the lysosomal content, there is also a progressive increase in the lysosomal delivery to mycobacteria in vivo. And that is something we can see here. In case of a wild-type mycobacteria, if we look at the survival in vivo, it follows a certain linear path. But if we take a mutant bacteria like a PKNG mutant here, which is delivered to lysosomes, it initially does not grow. but later on starts proliferating in vivo. So the lysosomal delivery is actually something that is not totally detrimental to mycobacteria, but I think it is arresting the growth of the bacteria, but not sufficient to completely eradicate. This is also something that has been shown in a different model of marinum infection in zebra fish. And very interestingly, the alteration in the lysosomal milieu is something that is particularly important for mycobacterial survival and trafficking. So here's an experiment where we infected mice with mycobacteria. And when the bacteria are still not in the lysosomes, we do a reinfection. And when we look at the trafficking of the second infected bacteria, it is still not in the lysosomes. But when we alter the trafficking milieu to such an extent that the bacteria are actually ending up in lysosomes, which is at a certain point after infection, you can see that the reinfected bacteria, which is very early in infection of the second infection, they are readily delivered to lysosomes. So the trafficking milieu is having a very significant impact on the mycobacterial survival. And we look more in detail about how exactly does mycobacteria modulate lysosomes. So based on the experiment from the David Russell's group that I showed earlier, where the surface labels of mycobacteria was seen all over the macrophage, we screened different mycobacterial surface components, particularly lipids, individual lipids of mycobacteria. And we saw that different lipids alone, when in the purified form, was sufficient to induce the biogenesis of lysosomes, like you see here with SL1 or with PIM6. And we worked out the signaling pathways by which these lipids, particularly SL1, is influencing lysosomal biogenesis. It's through the canonical mTOR pathway. But there are also examples of other lipids which are inducing biogenesis by probably non-canonical pathways that I'm not going to talk about today. So this is in the context of purified lipids. What is also interesting for us is we see that just by adding this lipid, there is an increased biogenesis of lysosomes. So what is happening is that when we simply increase the content of the lysosomes in the cell, you can see that the maturation of an independent cargo like E. coli shown here is increased. So here are macrophages which are simply treated with this mycobacterial lipid, but the maturation of a totally independent cargo like E. coli is very high in these cases. So mycobacterial lipids are able to both increase the lysosomal content and modulate the trafficking pathway of the macrophages, right? So this is all with purified lipids. What is happening in the context of the actual mycobacterial infection? So to address this, we took a mycobacterial mutant that does not make sulfolipid. And as we can see here, the mutant mycobacteria are able to show actually an attenuated lysosomal rewiring. There is much less lysosomes with the mutant mycobacteria compared to the wild type mycobacteria. And in fact, what is also interesting is that when we look at how much of mycobacteria are colocalizing with the lysosomes, remember, mycobacteria arrest the maturation of the arrest the phagosome maturation. So normally about 40% of mycobacteria end up in lysosomes, but in the case of the sulfolipid mutant, even less mycobacteria end up in lysosomes. So the arrest is much stronger in the case of mutant mycobacteria. And in fact, it correlates beautifully with the survival of the mycobacteria. You can see here that the mutant mycobacteria are actually surviving much better than the, it's surviving significantly better than the wild type mycobacteria. So clearly there is an association between the lysosomal induction of mycobacteria, the amount of lysosome, and the ability of the mycobacteria to arrest the maturation, and that is tied up to the survival of the mycobacteria, right? So this is about the survival part. What about the uptake part? So we were also looking at what is the determinant of mycobacterial uptake. And here again, we were looking at the endocytosis system. And there is something very interesting observation that came from Kuldeep in the lab, where we pre-labeled the cells with dextrin. So the macrophages take up dextrin with a certain distribution. And what Kuldeep saw was that it is in fact the cells that have a higher capacity for endocytosis that also take up more beats in this case. So here you can see cells that are sorted for higher dextrin versus lower dextrin. It is the cells with higher endocytic capacity that take up beats. And these are also the cells that preferentially get infected. So infection in a population is not a random event, but cells are having a certain predisposition for infection. What is also very interesting is that this endocytic capacity correlates nicely with the lysosomal content through mechanisms we don't understand very well. But also what is very exciting for us is that in an unperturbed cell population, we can see that there are distinct subpopulations of cell which are having a differential capability for mycobacterial trafficking. So we are now very interested to see if and how this correlates with downstream mycobacterial survival inside these different cells. So what I told you is that the heterogeneity in the endocytic capacity of mycobacteria is influenced the uptake, and the lysosomal rewiring mediated by mycobacteria is something that is modulating the bacterial survival. So what we are doing with this now is to do targeted chemical screens to identify molecules which are modulating the endolysosomal pathway. And we are having some interesting results in the lab which are ongoing, where we can see that by modulating this endolysosomal pathway, we can tune the survival of mycobacteria both to increased mycobacterial survival or reduced mycobacterial survival. Right, so that's, I think, the balance that is operating in the interface of the host and the pathogen. And as I said, we can have molecules which are both shifting the bacteria towards a lower survival or higher survival. And in fact, we see that this is having a corresponding effect also on the lysosomes. Right. So this is also leading us into the direction of heterogeneity in infection, which is a very exciting emerging area. If you can see, when a pathogen is infecting a host cell, a variety of different outcomes are possible, such as a cell does not get infected at all, or it can clear the bacteria, or it can get overwhelmed by the infection and it can die. The bacteria can survive in a certain persistent form, or it can start replicating. What is really exciting is that all of these can and do happen at the same time in the same cell population. So the outcomes of infection at a single cell level are distinct, but somehow they come together to influence a population outcome. Now, we all know that if you give a certain MOI of infection and look at the infection after a certain number of days, you're going to end up with more or less the same number of bacterial survival at the population. How do these individual cell outcomes influence the population outcome is something that is very exciting for us. And there are two models proposed in literature. One is the induced heterogeneity, which is heterogeneity that is induced by the infection, or a pre-existing subpopulation. So our own results that I briefly shared with you today is strongly showing the possibility of a pre-existing subpopulation with a differential susceptibility for infection. So how do we start approaching this? One of the emerging approaches is to start looking at the dynamics of infection events at a single cell level and follow it over time, like shown in this paper here, where they can trap single cells inside these microchambers and observe the interaction of the bacteria and the host cells. And here they can quantify various different types of outcomes. So our own analysis, our own approaches are showing a similar results. Here we can follow infection of macrophages with mycobacteria at a single cell level, and we can start observing the dynamics as well as the going subcellular and looking at the association of mycobacteria with lysosomes at a single cell level over time. How does it change? Does it change, etc. Right. So we are also now trying to observe some of these concepts and its relevance in the context of an in vivo infection. And for that, we are using different types of reporter strains of mycobacteria, which are giving us the physiological status of mycobacteria. So, for example, here is a lung section which is infected with mycobacteria. You can see different granulomas here, but the kind of association of these granulomas with different host cell markers, particularly the endolysosomal markers, are different. You can see here an entire granuloma associated with a certain host cell marker, LC3 in this case, and this granuloma not. So how does that influence the mycobacterial physiology per se? So these are some of the exciting questions. And going back to Rakesh's point earlier on AMR, Our results are also pushing us in the direction of host-based therapeutics, where we think by modulating host cellular processes, we will be able to influence mycobacterial infections. I briefly showed you glimpses of this by where we can tweak the host pathway and influence mycobacterial survival both ways, both more bacteria and less mycobacteria. We are gearing up also to do, we have been doing chemical screens to look at compounds which are targeting the host processes and killing mycobacteria, as well as there is a whole bunch of mycobacterial targets which are gaining relevance only inside the host, which is something that we discussed in this article here. So you can look at a directly relevant mycobacterial target with a host-based screening platform. That is something that we are gearing up to, that we are doing actually. So I will stop with this quote from Robert Koch, where he told very nicely in 1905 that if those tuberculosis people, and by that he means people who have the infection, but people who still go about their daily lives, they are abandoned to the fate, then a great gap in the battle against tuberculosis will result from this. So this was 1905, and this gap still exists. And I think it's important to remember this on the World TB Day, that TB still remains the leading infectious disease killer, apart from COVID, but it is still the leading killer with a single most infection with bacteria. There is the great gap that Koch mentioned is still, the gap remains, and we really need to fill this gap. And the new targets and new drugs are going to come from this gap. And I will stop with just one more point, that a lot of efforts on drug discovery are focused on directly killing the bacteria. What is also emerging in the recent years is that we need to understand the mechanisms which are restricting the growth of mycobacteria, not necessarily killing, but growth restriction, or factors which are controlling or constraining the growth, or the kind of host environments that are providing this constriction. Understanding this is really important to fill up this great gap that Koch mentioned. So I will stop here, and thank you very much for your time and attention.
Thank you, Vardha, for very nice presentation. I mean, it's obvious that we have to use multi-pronged approach to hopefully overcome this bug which has been bothering the humanity for centuries. And so you can stop sharing your presentation, Vardha. So there are questions accumulating in Q&A box. So I request the speakers to take a look if they can type some of those. Otherwise, we will, as I said, address them towards the end. Our next speaker is Dr. Sadhna Sharma. Professor Sadhna Sharma is head of the Department of Biochemistry in PGIMER Chandigarh. Her area of interest are biochemical and molecular basis of mycobacterial infections, nanomedicines, drug delivery, etc. Her research work is focused on understanding of various molecular mechanisms involved in the pathogenesis of tuberculosis and its associated diseases. Development of nanotechnology-based drug delivery systems capable of overcoming limitations of conventional therapy of various forms of tuberculosis is being extensively studied in her lab. Her team is also exploring the association of TB with diabetes and other complications. She's also working on targeted delivery of anti-tubercular drugs to the brain and their therapeutic efficacy against experimental tuberculosis meningitis. Professor Sharma, you can please start your presentation. Thank you.
Thank you, Dr. Rakesh, for a very nice introduction. So I think my talk will be slightly away from what have been done by the prominent scientists. So it is regarding the implications of nanomedicine based therapy for tuberculosis. So as said earlier, so tuberculosis is a very deadly disease, and as per WHO Global Tuberculosis Report of 2021, around 10 million people have been affected by this deadly disease, and the important risk factors which are attributing to this deadly disease include HIV infection, malnutrition, diabetes, smoking, and alcohol consumption. And in spite of the fact that very effective chemotherapy is available for the treatment of tuberculosis, still the incidence of tuberculosis disease is really going high. So what are the possible reasons for this? Because this existing chemotherapy is associated with number of limitations, which mainly include low stability of the various drugs in the gastrointestinal tract, then premature degradation of the drugs before they reach the actual target site, and most important one is the hepatotoxicity, which ultimately leads to the patient non-compliance and leading ultimately to the multi-drug resistance tuberculosis. So what are the basic solutions by which we can improve the existing chemotherapy of tuberculosis? So most importantly comes the nanomedicine approach. So this is basically a very promising approach which can really improve the drawbacks of conventional chemotherapy of this disease. And this
Mrs.
nanomedicine,
Sharma, can you go to presentation mode? I think your slides are not moving.
are not moving.
Yeah, we're still seeing the first slide. So maybe you can unshare and then share again. Yeah, now it has moved, but it's not in presenter mode. It is still in the viewing mode.
Okay. So now is it moving?
No, it is still.
No.
I would request you to once unshare and then share it. I think then it will come because it's the, yeah. Right.
No.
Yeah, now it's good. Yeah.
Okay. So
Yeah.
So here I was. So
Yeah.
I was talking about the nanomedicine, which is a quite interesting and promising approach, which can really reduce the main limitations associated with the conventional chemotherapy of tuberculosis. So it can overcome the limitations like the which includes the poor water solubility of the drugs, its low bioavailability as well as drug toxicity. And these nanocarriers, they have the ability to bypass the blood-brain barrier, the reticuloendothelial system, which normally affects the which affects the drug delivery. So ultimately, all these nanomedicine-based approaches, they have a immense impact on the healthcare, and it can really improve the drug development. So if we think why this nanoparticle-based approach is really important for the tuberculosis, so it might help to increase the therapeutic index of various drugs. It may help to increase the exposure of the target tissues to number of drugs. And most effective application of this nanomedicine-based approach lies in the chemotherapy of diseases which requires a long-term treatment. And in this treatment disease, we have a tuberculosis, which is a chemotherapy of which includes 6 months to 1 year. And it is mainly associated with the number of drawbacks, which most importantly includes patient non-compliance and hepatotoxicity. So till date, a number of carriers they have been utilized for the delivery of anti-tubercular drugs, which include macromolecular carriers, lipid-based carriers in terms of liposomes, then various polymer-based carriers, which utilizes both synthetic as well as the natural carriers. So we started our work with using both natural as well as the synthetic nanocarriers, and some of our earlier studies, they have indicated that when these anti-tubercular drugs, mainly the isoniazid and rifampicin, they were encapsulated in the various natural or synthetic polymers, they have a equivalent efficacy as compared to the free drugs, but and at the same time there was a great dosage reduction. That means the frequency of dosage administration was much lower as compared to then the free drugs are administered. So with this thing in mind, like we focused on another form of the tuberculosis, that is the extrapulmonary tuberculosis. So in this case, the tuberculosis meningitis is very important because it constitutes about 9.1% of the extrapulmonary tuberculosis. One third of the patients, affected person, they ultimately die from this disease, and 25% fatality they are in the HIV negative patients, and 67% are there in the HIV positive patients. So this is a disease which is having highest mortality among all forms of the tuberculosis. And the chemotherapy of this disease, it includes isoniazid, rifampicin, pyrazinamide, and ethambutol, which goes for around 9 to 12 months. And initial phase of the study includes also the steroids, which includes prednisolone and the dexamethasone. So there are several threats which are associated with the treatment of this tuberculosis meningitis, which include toxicity, that is the drug-associated side effects, interactions among various drugs. Then most of the drugs, they are not able to cross the blood-brain barrier, and the high cost involved in the treatment, and the lengthy treatment schedule which is followed to fully cure this disease. So ultimately, all these factors, they lead to the poor patient compliance and thus the treatment failure. So ultimately, what we need to think is that we require a suitable drug delivery system which should be capable of delivering the drug at the right place in the body, at the right doses, as well at the right time. And accordingly, along with the drugs, we should include in the drug regimen a suitable adjuvant which should be used to overcome the inflammation, which is a very common problem associated with the meningitis. So we thought of developing an effective strategy using the nanotechnology-based drug delivery system along with the various metallomatrix proteinases inhibitors for the effective management of the EBM. So though there are a number of delivery systems which are available, we thought of using the polybutyl cyanoacrylate nanoparticles, which is basically a biocompatible and biodegradable polymer, and it is mainly used as a surgery glue in the clinic. So this particular polymer is capable of enhancing the blood-brain barrier by 3 to 16 fold. So this polymer, it was coated with another moiety, that is the polyethylene glycol along with the polysorbate AT to enhance their uptake by the brain. Because the addition of the polyethylene glycol, it will protect the nanoparticles from its being uptaken by the reticuloendothelial system, and it provides its gastric digestion. And the further coating with the PAT, it will absorb apolipoprotein E from the blood, and nanoparticles enter the systemic circulation. And the LDL receptors which are present on the brain, they will internalize these nanoparticles through the receptor mediated endocytosis. So when these nanoparticles were developed by the polymerization of this butyl cyanoacrylate, so which is ultimately converted to polybutyl cyanoacrylate, and further coating of these nanoparticles with the polyethylene glycol plus PAT, it resulted into the development of the nanoparticles. So these nanoparticles were characterized through the X-ray diffraction and by the FTIR, and ultimately the surface morphology revealed the nanoparticles which were encapsulating number of drugs like isoniazid, rifampicin, pyrazinamide, and dexamethasone. All they were in the nano size range, ranging from 188 to 230 nanometers, with a varied degree of drug encapsulation as well as drug loading. So first, their internalization by the cells, it was done through the ex vivo experiment where the glioma cell line was used to study their uptake profile, and it was seen that fluorescently labeled nanoparticles, they were capable of being internalized by the C6 glioma cells, indicating that these nanoparticles, they have the potential to be taken up by the brain cells. So, and the coated nanoparticles were internalized at a much higher rate as compared to the uncoated PBCA nanoparticles. So then through in vivo study, we demonstrated that these nanoparticles, they can be taken up by the different organs of the animals, like when these nanoparticles, they were administered through the intravenous as well as through the oral route, and their uptake was studied at different time intervals. We found that these nanoparticles were observed when observed through the fluorescent microscope up to 96 hours, and both through the oral as well as the intravenous route, and the uptake as revealed by the fluorescence was much higher as compared to the uncoated nanoparticles. So free rhodamine could not be detected beyond 2 hours, whereas the coated rhodamine nanoparticles could be detected up to 96 hours, both through the oral as well as the intravenous route. And because it is very essential to study their pharmacokinetic and pharmacodynamic component, so we studied these two parameters through the three formulations were taken into comparison. One is the free drugs, another one is the uncoated nanoparticles, and another one is the nanoparticles which have been coated with the polyethylene glycol and PAT. And these drugs were given at a therapeutic dosage. And it was observed that as compared to the free drugs, these drugs were found in the circulation up to 168 hours as compared to the free drugs which were cleared from the circulation, not they were cleared by 12 hours only. So that could show that these nanoparticles, they have to have the ability to release the drugs in a sustained manner. And interestingly, these drugs were capable of entering into the brain, as we can see that the brain has a quite high concentration of the drugs which have been released from the nanoparticle. So this holds true for the isoniazid, rifampicin, pyrazinamide, and the dexamethasone, which is an essential component during the initial phase of the therapy. So their pharmacokinetic and pharmacodynamic parameters were significantly improved, as we can see from the high Tmax and the area under curve as compared to the free drugs and as compared to the uncoated nanoparticles. Again, emphasizing the fact that these PBCA nanoparticles, they have the ability to release the drug in a sustained manner. So they have the capability to reduce the toxic effects which are normally associated when the drugs are administered at once. So these nanoparticles, they had enhanced and sustained drug delivery to brain. There was prolonged retention of drugs in plasma and various target organs, and the drug concentration was relatively high in the brain as compared to rest of the organs. So as I said earlier, that inflammation is the most important complication which is associated with this particular disease. So this inflammation is associated with because of the high concentration of the matrix metalloproteinases. So which ultimately leads to the increased cytokine production, and ultimately it leads to the neurovascular matrix degradation, as well as it disrupts the blood-brain barrier, which ultimately leads to the cell death, neurotoxicity, and the brain hemorrhage. So we wanted to see that if we can somehow inhibit the MMP9 levels by incorporating the particular inhibitor of the MMP9 along with the various anti-tubercular drugs, whether it can avoid the complications which are associated with the with the disease. So this matrix metalloproteinases, they are basically the endopeptidases. They have the ability to degrade the extracellular matrix. They are involved in the cell proliferation, migration, and apoptosis as well as. Though there are different types of the MMPs which have been discovered so far, but the most important one which is there in the TBM is the MMP9, which is known to disrupt the blood-brain barrier in number of central nervous system disorders. So its high levels have been demonstrated in the multiple sclerosis, meningitis, and brain tumor. So the increased level of this particular MMP, it is associated with the decreased levels of tissue inhibitor of metalloproteinases 1. So we need to keep a balance between these two, that is its inhibitor as well as its activity as such, to have the normal blood-brain barrier functions. So our purpose was that we need to select a MMP9 inhibitor which could decrease the levels of MMP9 to maintain the integrity of the blood-brain barrier. So initially, like we just wanted to see that whether there is really an increase in the MMP9 levels in the CSF, which is obtained from the TBM patient, and we demonstrated that there is increase in the MMP9 levels as the disease get advanced. So in the third stage of the disease, there is a significant increase in the MMP9 level. So it was demonstrated both in the CSF as well as in the blood. So we can say that it is really important to take care of the high levels of MMP9 to overcome the disease complication. So for this, we procured a specific inhibitor of the MMP9, so which specifically binds to the zinc ions, thus reducing the activity of MMP9. So when this inhibitor, it was given along with number of anti-tubercular drugs, we found that there is a significant decrease in the mycobacterial burden when the C6 glycoma cells, they were infected with MTB S37RB. So indicating that a reduction in the MMP9 level could really decrease the mycobacterial burden in the cells. Similarly, it was further supported by the finding that this inhibitor is really capable of reducing the MMP9, which was again demonstrated through the zymographic analysis of the MMP9 levels in the infected cells. So our observation revealed that MTB infected cells, they demonstrated increased MMP9 levels, and its treatment with the MMP9 inhibitor, it could decrease the MMP9 levels. And anti-TB drug treatment alone, it is not effective for the enhancement in the MMP9 levels, but if it is used along with the dexamethasone or with the specific inhibitor of MMP9, it can really decrease both MMP9 levels as well as the bacilli burden. So these coated nanoparticles, they had the ability to enhance the bacterial clearance as well as to inhibit the MMP9 levels. So further, after doing these studies in the cell lines, the same findings, they were confirmed in the animal model where the infection was established by giving the MTB S37RB to the mice, and infection was established after 4, 8, and 12 weeks of infection. And it was found that there is a consistent, constant mycobacterial load in the brain, indicated that the infection has really established in the brain. So at 4 weeks of the establishment of the infection, the treatment was started. And again, the establishment of infection was demonstrated by the histopathological examination of the brain, which showed tissue inflammatory lesions in the brain, followed by the edema of the brain tissue, indicating that the bacilli has really established the infection in the brain. So after this, the chemotherapy was given for 4 and 8 weeks, and their mycobacterial burden was assessed in different organs and followed by the histopathological examination as well as by the MMP9 levels. So as we can clearly see over here, that as compared to the untreated brain of the rats, of the mice, there is a significant drop in the CFU, which have been CFU in the animals which have been treated with the PBCA nanoparticles encapsulating different drugs as compared to the uncoated nanoparticle or as compared to the free drugs. So the point to be noted over here that the free drugs, they have been given daily, whereas the nanoparticle drugs, they have been given after every 8 days. So there is a 8 times reduction in the frequency of the drug administration. So this is the most salient finding of our study. Similarly, these findings, they were supported by the histopathological examination. So as compared to the untreated animals, which indicated a heavy meningitis as well as the inflammation, after the treatment with the nanoparticle which contained anti-TB drugs, there was reduction in the inflammation, but it was not completely eliminated, indicating that the introduction of the nanoparticular therapy might be much better as compared to the free drug therapy. Similarly, the findings were further supported by the decrease in the MMP9 levels in various organs like brain, lungs, as spleen, as well as the serum. So these coated drug nanoparticles, they demonstrated accelerated bacillary clearance. And as I said earlier, that there was dosage reduction to the 1/8 of the original dosage. And free organ nanoparticulate drugs, they were not very much effective in decreasing the MMP9 levels. It is only effective when they are given along with the inhibitor of the MMP9. So this is case when this conventional chemotherapy of tuberculosis is given along with the specific MMP9 inhibitor. So there is a significant decrease in the CFU in the brain as compared to the untreated animals. So similar findings, they were demonstrated in terms of the histopathological examination. So the brain could achieve the normal brain histology after 8 weeks of the chemotherapy. So overall, what we can say that when the animals, they have been infected with M. tuberculosis, which results into the tubercular meningitis, as demonstrated by the mycobacterial burden seen in the brain. So which is revealed by increased MMP9 levels, inflammation, as well as the tissue destruction. So when these animals, they were given the anti-TB drugs along with MMP9 inhibitor, and these drugs, they have been encapsulated in the nanoparticles, it resulted into the sustained release, which ultimately resulted into the treatment of the disease, and particularly the high drug levels have been demonstrated into the brain. So we can say that these nanoparticles, they are quite targeted towards the brain. So which is because of the decreased MMP9 levels, decreased inflammation, as well as tissue destruction. So this is the overall work which we had carried in the direction of the tuberculous meningitis. So simultaneously, the work has been done by using various other drugs as well, as like number of azole drugs, then phenothiazine, and currently clofazimine and verapamil has also been used. So I think this is what we have been done for the nanomedicine based approach for the treatment of tuberculosis. So this work was possible by the funding from the DST as well as from the Indian Council of Medical Research, and this is the effort of my various co-investigators, which are from the Department of Medicine and Neurology, as well as various PhD students. So with this, I would conclude my talk. Thank you.
Thank you, Professor Sharma, for giving a very detailed description of what are different ways of handling this pathogen. And I'm sure various kinds of drugs, host response, we have to use all possible tools to eradicate tuberculosis as our country is aiming at. So, yeah, thank you again. And so we will take up questions after now the second talk, the last talk. There are many questions that are already answered. So I request audience to keep typing their questions in the Q&A, and we'll take care there. Our next speaker is Dr. Urvashi Singh. So Dr. Urvashi Singh is a professor in the Department of Microbiology at All India Institute of Medical Sciences in New Delhi. She's also chief of the tuberculosis section at AIIMS, and she's an active member of various committees on tuberculosis and works with the Government of India on treatment, prevention, and diagnostics of TB. She served as a member of editorial board of Frontiers in Microbiology and various other national and international journals. So we are going to get yet another perspective of the tuberculosis. So Dr. Singh, you can start your presentation. Thank you.
I must thank the organizers at the outset, Dr. Mishra and the team. So I would like to, at the outset, say that I was asked to cover the diagnostic challenges. So I may not be really able to discuss our work like the previous esteemed speakers have been able to do. And this perspective, like Dr. Mishra said, it's a totally different perspective. But we're going to talk about how all the research, the molecular research that is being talked about, can actually be translated into patient treatment decisions. So going ahead, I would like to So a brief glimpse of the global burden. Dr. Sharma talked about 10 lakh cases. India has reported about 9 cases per lakh population. And I also want to bring in the National Drug Resistance Survey, which was done a few years back. But it is of relevance to what I'm going to bring today to everybody's attention. The resistance to INH, though I'm not sure how many clinical people are here, but the message I'm sure will hopefully be clear towards, as I cover the different aspects of the mycobacterial biology and its relevance to clinical treatment. So the national DRS observed any H. By any H, we mean whether it was a mono H resistance or it was a poly H or it was as part of MDR, which is defined as resistance to both INH and rifampicin. So 11% INH resistance was found in new or drug-naive TB. The patients were reported for the first time to the program, and 25% in previously treated TB. And these were the drivers of rifampicin-resistant TB, which is the most difficult to treat TB, needs prolonged treatment, needs different regimen combinations, different drug combinations in regimen, and still a lot of patients fail. So slides are a little slow to move. I cannot see why. So this is the brief of what I mentioned earlier. The INH resistance was found to be 11%. And if you look at the quinolone resistance, was also equally high. Additional quinolone resistance to MDR-TB patients in MDR-TB patients was to the tune of 21%. And for second-line injectables, it was to the tune of about 3 to 4%. Now, why all this is important is you will see that when the regimen has been laid down by the program, the relevance of these drugs comes up. And if you look at the WHO recommended regimen, these drugs are very, very important in the treatment of MDR-TB. Before I move on to the recommendation by the WHO and the National TB Program, I want to bring to your attention this article, which is published recently in 2022, which talks about mortality in MDR-TB patients and a comparison of routine drug susceptibility testing with the whole genome sequencing. The study was conducted in 7 countries where they showed that of 582 patients with tuberculosis, 58% isolates were pan-susceptible, 6% were monoresistant, that is resistant to only one drug of the drugs used for treatment of MDR-TB, and 25% were multi-drug resistant, with also a pre-XDR number of 4%, and XDR, actually 4% were pre-XDR, XDR together. So when they analyzed the mortality on these 530 patients, in all, 12% patients died, 15% patients received inappropriate treatment. This is in total. Mortality ranged from 6% in patients with pan-susceptible TB to 39% in patients with pre-XDR and or XDR TB. This was what they summarized. They recommended that whole genome sequencing would make the story a little better. And 32% in drug-resistant TB mortality is very high. what they also showed is that undertreatment was responsible for mortality and overtreatment did not harm many patients. So to the left of this graph is the mortality in pan-susceptible and monoresistant in MDR, and as is well expected, very high in pre-XDR and XDR. Also, comparison of mortality to discordance. By discordance, they mean between the phenotypic and the genotypic methods, or the whole genome sequencing and the phenotypic culture-based methods. Mortality was high where the discordance was high and the patient was treated using one of these methods. So the message is very clear that we need better methods of diagnosis and treatment. So this is a summary of the treatment success rate in India. Till about 2017, the success rate was never more than 50%. It was the introduction of bedaquiline under CAP in the year 2017 that the success reached 71%. And since then, different regimens. So we've had a shorter MDR regimen where the outcome has not been very great. Again, thanks to the prevalent drug resistance in the country. And with regimen, with a few other regimens, the outcome has been a little better. So this has been the classically shown as the change in drug resistant profile in our country, 2 different first-line TB drugs. This is the summary of the regimens recommended now by the National TB Elimination Program. This was released around the TB day. So for drugs susceptible TB, we use first 4-line drugs for the intensive phase of 2 months, followed by 4 months of HRE. This is where the INH resistance that I talked about earlier comes in. So a few years back, the continuation phase only consisted of H and R, and E was introduced as a rifampicin protective drug. When the NDRs, etc., became available and we knew the baseline H resistance was very high, for only H resistance, either mono H or poly DRTB, the regimen recommended includes levofloxacin, which is a quinolone, along with rifampicin, ethambutol, and pyrazinamide for 6 months. Now, of concern here is the use of levofloxacin and keeping something like 20% plus drug resistance to quinolones in our country. This again is a cause for concern. For treatment of MDR, rifampicin resistant and XDR-TB, what is recommended is in the shorter regimen, we have about 9 to 11 months of shorter regimen where we use bedaquiline for 6 months, 4 to 6 months, depending on the patient's outcome or the response, along with levofloxacin, clofazimine, pyrazinamide, and ethambutol, high INH, ethionamide, or 5 months of levofloxacin. This is the continuation phase where we have 4 drugs, levofloxacin, clofazimine, pyrazinamide, and ethambutol. The injectable regimen, which comes in the next row, has been totally asked to be removed. So there was a lot of pressure from evidence put together by WHO, several studies they put together, and the benefit was not as much as the harm due to the injectables, amikacin, kanamycin, and capreomycin. The longer oral regimen includes levofloxacin, bedaquiline, linezolid, clofazimine, and cyclosidine. Bedaquiline and linezolid are given for the initial few months, and the linezolid dosage is tapered as required. We're also coming up with TB preventive treatment, 6H, which has been given traditionally for years together. And I'll come later that this could have been the reason why we have higher INH resistance in our community. Now what is being recommended is INH with rifampicin for 3 months of 12 dosages, that is once in a week dosage. Levoflox for the contacts of MDR and rifampicin resistant TB patients. The other option is 4 months of daily rifampicin in contacts of H resistant but RIF sensitive index patients. So this is the integrated management algorithm for TB disease and infection. So what is, I would like to just quickly run through these slides to just share with you the methods used for diagnosis of drug resistance. Recommended are nucleic acid amplification test, microscopy and chest X-ray, of course, and the line probe assay along with liquid culture DST. So the ones which are highlighted based on all, using all these tests to diagnose the resistance profile first to rifampicin, because that decides whether rifampicin is going to be the mainstay of the drug treatment, or we will need to add, other drugs such as second line drugs. So this is appears to be the slide appears to be very, very complicated, but what we are doing here is we are understanding the presence of mutations in the strain infecting the person, and depending on the presence of resistance to a certain drug, and also we also like to know the level of resistance, whether it is low level resistance or high level resistance to a few drugs. We can substitute higher drug dosages to overcome low level resistance. For example, the classical example being INH and fluoroquinolone, especially moxifloxacin. I will detail this later. For rifampicin also, high dose rifampicin has been used in certain countries with very good outcome. But in our country, we continue to use the standard dose of rifampicin. So I'll move on. And this is another slide which talks about how the program recommends the schema of choice of whether we use a shorter oral bedaquiline containing regimen or longer oral MDR XDR TB regimen based on the presence of, so in case there is no additional resistance detected after rifampicin resistance detection, and if the INH resistance detected has either CAG mutation, which goes hand in hand with high level resistance to INH, or INHA mutation, which is responsible for low INH resistance, but is responsible for ethionamide resistance. So the reason why we do all this is because the drugs in the regimen have to be at least 6 to 7 in number. So if we have CAG mutation, we lose a high INH, which cannot be used here. If we have INHA mutation, we have ethionamide, we will lose ethionamide, so we will not be able to actually give the shorter oral regimen complete. Now, the relevance of the regimen and the baseline sensitivity to the drugs being used is very, very important. And I bring in 2 or 3 cases towards the end of the talk, which will highlight why it is very important to know what drugs cannot be used. Longer oral regimen we can use in fluoroquinolone resistance patients, and in which both CAG and INHA mutation is detected, so we can do away with high INH and ethionamide. Now, you may be getting bored of too many flowcharts, so I don't think this has many new messages, and more or less I have talked about the diagnostic methods. All of you are familiar with microscopy. So this is the initial test which was being recommended in the peripheral areas, but of late we have had very good nucleic acid amplification tests which are available and have been tested in the peripheral areas. The picture of LJ colonies which take longer, 4 to 6 weeks. This is the liquid culture system, MGIT, which is considered as gold standard, results within 2 to 4 weeks, and we can do DST following the phenotypic DST can be done. In the phenotypic DST, the critical concentration is defined for different drugs in different media, and this will tell us if the organism is resistant to the critical concentration. For certain drugs, in addition to the critical concentration, we have higher concentration, which can be, so the drugs which are able to achieve a higher drug level in a patient, for example, moxifloxacin, can be tested at a higher concentration for the use of high moxi, similarly for high H, and similarly for high rifampicin. Now, before I move on to the molecular tests, I wanted to briefly bring in the concept of molecular Cox postulates. Of course, what we are using in diagnostic is, so this is a slide which talks about the turnaround time for different tests. The basis of the GeneXpert test uses molecular beacons, which the probe, if it binds, will release the fluorophore, and we will have the emission of light. This is how the real-time PCR in the Expert G4 cartridge, which was the first cartridge introduced in 2011, will indicate presence of resistance due to non-binding of a probe of the 5 probes in the RPOB region, which is responsible for resistance in rifampicin. So this is a TrueNAC test, which is a two-step test. We do DNA extraction followed by amplification on a chip, and the result is indicated as MTB detected and followed by resistance. This is a new cartridge by GeneXpert, which is the XDR cartridge, uses 10 colors and uses HRM technology. The TrueNAT also uses HRM technology, and these are the targets which are covered in the XDR cartridge: the CATG, FABG1, OxyR, AHPC, intergenic region, INH promoter, which is responsible for low-level resistance to INH, but ethionamide resistance completely. Fluoroquinolone, they cover GAIR-A and GAIR-B. For the injectables which are no longer being recommended, they cover the RRS and the EIS promoter. So nevertheless, though they're using the old definition of XDR, which is resistance to both quinolones and injectables in addition to MDR, but this cartridge is still very useful to us because at the outset we can know whether the INH can be used or not, whether ethionamide can be used for the regimen, whether we take the shorter regimen course or we take the longer regimen. Quinolone, which is an essential component of mono-H treatment, the shorter MDR regimen, the knowledge of resistance to quinolones becomes very important. And this is the sensitivity as claimed by them. Ethionamide, since they're covering only one gene and not the other genes responsible. And I will also briefly touch upon which gene targets have been defined for which drugs. Now, this is how the XDR cartridge results will show up. We have done an initial study, and this works very well and it has been of a lot of aid to our patients who are failing on a bedaquiline regimen. So we are able to know which are the other drugs which we cannot use. So we obviously can tailor the regimen as per the needs of the patient. Now, briefly touching upon the line probe assay, which uses reverse hybridization on a solid phase on a nitrocellular strips, we can detect the presence or absence of wild type or mutant probes. And this is a real time, this is a picture of a test done in the lab where you can appreciate the mutant probe in the GAIR-A has been picked up. And there's been a loss of wild type probes in this particular test. So this indicates mutation in the that particular site of the mutant probe. This actually goes hand in hand with high fluoroquinolone resistance. Now I come to the challenges that are faced due to disease biology. So this is I'll run through this quickly. This is the initial infection contained. If it is contained by the innate immune response, there will be no CMI to follow up if we look for presence of any latent TB infection. But in case the adaptive immune response kicks in, we will have a CMI which is detected by TST and IGRA tests later. The primary TB is contained because and in the post-primary TB we can see there is a reactivation of there's granuloma expands, leads to capillary formation. So what I want to bring in here is what Dr. Varada also showed, that this is a cyclical disease, quite dependent on the immune response of the patient and of course the virulence of the pathogen. So there's a balance between the two, and we are now, this is a recent concept. For the past 5 to 10 years, we are now discussing not the two ends of the spectrum, latent and active. We also realize there may be incipient and subclinical TB, and the differences are very subtle, really. So we may have culture positive intermittently in subclinical TB disease, which is always positive in active disease. Though in latent, we're not able to grow culture, but the replication does go on at a lower level. So this is another table which talks about the similar. So I wanted to actually bring in the challenge here. So what is the challenge? What has been proven? This paper was published by Ford et al. In 2011, where they showed that MTB acquires a similar number of chromosomal mutations during latency as occurs during active disease, indicating thereby, and they also observed that the MTB continues to acquire mutations during latency and provide a novel explanation for the observation that INH monotherapy for latent TB could be a risk factor for emergence of INH resistance. And that is why the picture I showed earlier, where I showed that the resistance has been reported in higher numbers and the NDRS is clearly shown. And this was the reason the treatment was changed. The continuation phase now has 3 drugs instead of 2 drugs earlier. Now, moving on to drug resistance in TB, the classical factors which have been described for MDR-TB are delays in diagnosis, no access to diagnosis, or drug susceptibility testing, delay in communicating test results, poor sensitivity and specificity of diagnostic methods. I have narrated the ones recommended by the program currently. Of course, they're moved from what were the previous recommendations. Treatment regimens, if they are inadequate, if the drug supplies are interrupted, poor drug quality, drug-drug interactions, which could lead to lower levels of certain drugs. Lower level of drugs are known to enhance drug resistance because they will allow mutations to go on. And I will briefly touch upon which mutations are responsible for drug resistance in the next few slides. Also, patient-associated factors like high-risk living environment, behavioral factors like non-compliance, treatment-seeking behavior, etc. Patient-dependent pharmacokinetic and pharmacodynamic properties, other immunological factors, comorbidities such as HIV and diabetes. Now, the pathogen factors such as persistence, Dr. Varada briefly mentioned, but of course, persistence is now being investigated in a big way. Phenotypic variability of bacteria, dormancy of bacteria, increased efflux pump activation, which is described to occur in lower drug levels when the patients are given drug regimens in lower dosages as required. Other differential gene expressions, asymmetric cell division, biofilms formation. These are query because some of them there is evidence to and against most of these features. The genetics of the bacterium, there is high baseline fitness of strains. Now, this again I'm going to come to in the next few slides, that the fitness may be compromised due to certain drug resistance mutations, and then the compensatory mutations kick in to balance out the fitness, the lost fitness of the strain. Epistatic interactions and sign epistasis, again, I'm going to touch briefly upon. Elevated mutation rate could be one reason which generates MDR-TB, and there may be cross-resistance patterns, like I talked about a few mutations which can lead to cross-resistance. There are more, and there may be still more which are not yet described, and we will learn by and by.
Dr. Singh, can you wind up quick, because we are running out of time, so we need to also have question and answer sessions. So if you can quickly wind
up. Very well.
up.
I think I still have at least 10 salient slides. If you may kindly consider 5 more minutes for me.
This is going into question answer session. Yeah, please do it quickly as you can. Yeah.
Very well. So let me talk quickly about these are the mutations classically described for resistance to different drugs, and so the fitness and compensatory mutations kick in due to epistasis. Now, like I said, resistant bacteria may lose certain resistant mutations could lead to loss of fitness in Mycobacterium, and which may be compensated by a mutation at another site. Epistasis, which is actually defined as effective interaction between 2 or more mutations, can actually help stabilize. Now, acquisition of 1 or more additional resistance determinants may sometimes ameliorate the fitness cost of the first mutation, which is given the name sign epistasis. MDR strains could actually improve their fitness by acquiring mutations conferring resistance to additional drugs. Now, this is a very classical example. The CAT315 mutation, which leads to high INH resistance, is balanced out by mutations in the other CATG sites or in the AHBC gene region, which, if alone, leads to low-level resistance to INH. RPOB mutation, which is responsible for high-level resistance to rifampicin, is balanced out by mutations in RPO-A or C. Similarly, RRS1401 has a compensatory mutation supporting it in the other sites of RRS, and GAIRE94 has unknown mutations, compensatory mutations. One of the compensatory mutations that has been described, I'll come to the RPOB mutation, which is known to be, they are known to support each other. This is a picture of from a recent publication which talks about definite known compensatory mutation which balances out the fitness of. So positive epistasis may drive multi-drug resistance. And something of concern here is drug regimens containing both rifampicin and fluoroquinolones may need to be looked at again, because strains with specific combinations of Guide D94G and RQBH4455Y are fitter than the wild type, and they be selected out. They can be one of the more efficiently transmitted. Another picture to show higher MICs in epistatically balanced mutations. This paper by Sebastian Gagnon, published recently, has shown that compensatory mutations were noticed more in prison, inmates, and they were able to show a correlation. At least 60% of MDR-TB was due to patient-patient transmission, and compensatory mutations were responsible for a higher number in the prison patients. And epistasis, that is compensatory mutation, and the drug-resistant mutation also are affected by strain genetic background and could affect higher transmission, higher levels of MIC, competition with other strains. I just have one more concept I want to bring in, is the concept of polyclonal TB and heteroresistance. This is our publication in the year 2020, where we showed in sensitive TB as well, polyclonal infections were very high to the tune of 53% at the baseline, and the genotype changed during treatment while the patient was on treatment. So this questions the basic tenet of MTB, reinfection and reactivation. There is always quite likely, even if we could show this in sensitive patients, there's quite likely that the colony, the cavity is filled with more than one strain type. And this is a recent data during pre-COVID time. Our polyclonal infections were much less, and this is a post-COVID time where polyclonal infections were reported more. Now, what this brings us is to the concept of heteroresistance and emergence or amplification of resistance during treatment. This is an article by Sebastian Gagnon, where you can appreciate initial resistance to drugs 1 to 7 were there, but during treatment, the patient developed resistance to the 8th drug and later to another 9th drug. And there were variants, there were multiple variants observed in this host. And this is actually trying to show different detection methods have different abilities to detect whether 10% of population has is resistant or more. So certain tests will require 100% of the population of the cavity clones in the cavity to be resistant. Then only a test will pick up. Certain tests have better pickup limits of detection. Now, this is a study which is a meta-analysis which showed of 38 studies, heteroresistance rifampicin was reported in 17 studies, and in fluoroquinolone, heteroresistance reported in 12 studies, and for ethambutol in 4 studies. So heteroresistance is not so uncommon as maybe. This paper talks about bedaquiline resistance in a patient on treatment, and this paper in JBC talks about how bedaquiline could interfere with the action of INH and moxifloxacin, the antibiotic action of moxifloxacin and INH, because those two lead to enhancement of ATP intracellularly, while bedaquiline acts by curtailing the ATP. This paper talks about microevolution in a patient on bedaquiline regimen, and they show that the ATP mutation, ALA63Pro, which is responsible for resistance to bedaquiline, was absent at the baseline, detected 6 months after stopping unaffected treatment, and it vanished towards the end, and the only mutation they detected towards the end was the RB0678, which is responsible for an efflux pump and is shared between clofazimine and bedaquiline, and this is a picture which elaborates the same. This is another patient was reported in 2015, and in this patient as well, at the baseline, there was no resistance detected to the drugs used for treatment. This was a Nepalese patient treated at Switzerland. 3 papers were published for this one particular patient, one in AJR, CCM, and in 2 more, one in NEJM. So this patient acquired resistance to bedaquiline, bedaquiline as well as delaminid. This is the delaminid picture, AJR, CCM in 2016. And this shows what all mutations he acquired while on treatment. So this only indicates that you cannot use drugs like bedaquiline and delaminid on a failing regimen. So the understanding of the milieu of the cavity is very important. So the ideas I brought in actually question the age-old dogmas. So the age-old dogma was resistant strains are less fit than susceptible bacteria. There is evidence to the contrary. M. tuberculosis is clonal with no phenotypic or genotypic variation. Again, this has been questioned by recent evidence. We have questioned concept of reinfection and reactivation because it's always a mixture, whether you can detect it or not. Knowledge of bacterial genetics and evolution must contribute to treatment regimen as well as tuberculosis control policies. And the reason why we may be failing is not giving due acknowledgement to this knowledge and still continuing with the regimen from the drugs are chosen as it's a reassortment. You just pick and choose. Of course, you pick and choose based on the patient treatment outcomes. Databula is considered wonderful, but we have reports of
Dr.
resistance.
Singh,
being
Dr. Singh, we are
reported. Thank you.
really out of running out of time. Please
Very well.
conclude
Thank you
because
so
we have
much, sir.
other
Thank you
things.
to the listeners as well, and I hope it is useful.
Thank you. Thank you so much for very elaborate presentation. I'm sure it was liked by our participants. So now we will play the message from Dr. Soumya Swaminathan, who exclusively recorded for this purpose.
Greetings, colleagues, and all good wishes for World TB Day 2022. Thank you for asking me to say a few words about innovation and TB. I think we're at a very critical juncture because of the pandemic. TB and other disease control programs have suffered big setbacks. And as you know, the last year's WHO Global TB report showed for the first time that there was an increase in reported TB deaths and a significant reduction in notification of cases. We had gone to 7.1 million cases being notified 2019, and this dropped to 5.9 million. And at the same time, the number of deaths increased. So these are reported deaths. So it's in all likelihood, the real deaths could have been higher. And we know that we have still not been able to come back to the pre-pandemic levels of case detection and case holding and management. And at the same time, when we track the outcomes of TB treatment, while outcomes for drug-sensitive TB are very good with the existing regimens, 6-month regimens, the outcomes for MDR TB and XDR TB are still very far from being what we might consider optimal. And I think the latest data from India shows that less than 50% of patients are getting cured. And of course, there's a huge gap still in globally the detection and treatment of MDR TB as well as in preventive therapy. So what should we do? Because we have these very ambitious goals for India as a goal for 2025 of TB elimination. There are the 2030 goals, the SDGs, and then there are the Stop TB goals, 2035 and 2050. And I think we are nowhere close to achieving any of them. And let us see what data the TB prevalence survey from India shows. But I believe that it will show that prevalence is very high. So we need innovation. And where do we need innovation and how do we go about it? We need innovation in all aspects. As you look at the cascade of care, we have got used to thinking about it for HIV. We should think similarly for TB. In a community, we have X number per thousand who are likely to have TB, right? Of them, we detect X percentage. Of those diagnosed, X percentage go on treatment. And of those who are started on treatment, a smaller percentage are cured and a smaller percentage remain TB-free, recurrence-free. So ultimately, when you look at the percentage of people who are cured without recurrence, out of the total population of TB, it is small. It may be 30 or 40%. And so the majority of TB patients are not getting the care that they need. And so we have to look at the cascade, we have to analyze the data accordingly, and we have to think, what do we do to improve the early detection of TB? How do we capture all the cases in the community? For that, you need community engagement and involvement, and you need all of the NGOs and the activist groups and the ground level work that needs to get done. and you need better diagnostic tools. We did it for COVID. We should be able to do it for TB, rapid diagnostic tests. Then you need to come to the next step, which is starting every patient on the appropriate treatment. For this, we need to use the best available regimens. There is no point in using regimens which are now outdated. We know we BPAL for XDR and pre-XDR TB gives very high cure rates. There's no reason why we should not be using BPAL. Similarly, for drug-sensitive TB, we know now that a 4-month fluoroquinolone containing regimen is as good as 6 months. We've just seen data for children showing that a 4-month regimen, standard drugs, is as good as 6 months for uncomplicated TB in children. Similarly, for complicated TB, meningitis, etc., we know what drugs we need to use. So optimal management, for that you need early diagnosis and diagnosis of drug resistance. Then you go on to managing the patient, following them up, making sure there are no sequelae, and doing any rehabilitation or surgery if needed, etc., to give the patient the best quality of life. And before all of this, of course, comes prevention. And prevention either through vaccines or through, again, better preventive therapy regimens. We are not doing well on prevention. We need more research. We need more clinical trials. There are many vaccines now which are sitting in phase 2. They need to progress to phase 3 and beyond. I think this is the ideal time that India can take a lead in launching a number of clinical trials as well as a number of implementation research studies at scale to show that changes can be made in the way in which we do things. And again, we need to involve the TB patients, we need to involve the communities, and we need to make sure that the social determinants of TB, which are mainly poverty, undernutrition. In India, undernutrition we know is the biggest risk factor for TB. And then others like smoking and exposure to air pollution, etc., also need to be addressed.
Thank you. So with that, I'd like to thank all the speakers for various aspects of TB. And now I hand over the proceedings to my colleague, Dr. Harvinder Khera, and she will handle the question and answer session. Harvinder, you can go on.
Thank you so much, sir. And thank you, eminent speakers, for your insightful presentations. So we are moving to the discussion session. My first question is to Dr. Varadha. So Dr. Varadha, you presented really nice talk. So my question to you would be, can we develop some biologics for TB? For example, antibodies that can block viral the pathogen entry?
Yeah, so thanks, Harvinder. I think people have been looking at this for a very long time. The entry pathways are quite complicated. So there is no single receptor. There are multiple receptors known for entry of TB. So blocking one is not necessarily going to help us solve this. The other way people have been approaching is also to activate macrophages by a lot of cytokine mediated approaches with interferon gamma and other things. Again, it also goes back to the point made by Dr. Sadhana Sharma about the immune responses and the inflammation being a major player in pathologies, right? So pathologies is inflicted by the immune responses of the host system. So there are approaches along the way, but I think we are very far. The other biologic was also, I think, a question that was posted there on phage mediated therapy. And that is also something a lot of people are looking at. I think there are phages available that work fairly well in cultures. But again, we need to realize that the bacteria is hiding inside a compartment inside the cell. So whatever we do, a drug or nanoparticles or phage or biologicals, we need to have optimal way to get it there, right? And that is also a very big challenge. And just to conclude that thought, when this is all these challenges is looking at bacteria hiding inside a vesicle inside the cell, even in the context of a simple cell culture infection model, right? In the case of an animal infection model, you're only going to add that much more layers of complexities.
Thank you, Dr. Varadha. My next question goes to Dr. Sadhana. Ma'am, can you please tell what are the challenges that you face for diagnosis of especially the non-pulmonary tuberculosis cases?
Yeah, the main challenge which is being faced is like when the patient really comes to the hospital, he is already having the TBM, but the condition is really quite critical and the disease is being not diagnosed properly. So the complications of the disease go very serious, and ultimately the patient, that's why in the case of TBM, the mortality is really very high. So the diagnosis is the main challenge which is there for the diagnosis of TBM particularly. Thank you.
Thank you, ma'am. Thank you, ma'am. Dr. Urvashi, can you tell us, recently we have seen the pandemic has really impacted every sphere of life. So how do you think COVID has impacted TB and other respiratory disease diagnosis and treatment?
Well, this has a multi-pronged answer. So one is the social factors that led to delays in diagnosis, delays in treatment, which enhanced transmission within households. People say masks were used, but masks were never used inside their households, and people were actually avoiding the use of masks. So the transmissions went on. Patients with early signs of disease could never reach a center to get the right diagnosis and treatment. So there were delays in diagnosis, and so what we are noticing now is highly complicated disease. We're noticing patients who are reaching us have, if I put it in crude words, the lung shadows completely filled with the disease. So they probably, they had fear of not coming to the treatment center because they were afraid of COVID. So there were delays of all kinds, and like I always say, COVID has been a very good teacher. So like Dr. Samya also mentioned, if we have been able to deal with COVID, we should be able to deal with TB. But so the difference between COVID and TB really is the incubation period, and this kills right in front of your eyes, while TB will kill slowly. And people can probably never understand how severe the disease, the TB could be. Like Dr. Sadhana talked about meningeal TB, the mortality is so high, and one of the reasons why it is so high is because the treatment drugs are not reaching the right site of the disease. So her work will prove to be very useful if we could use nanoparticles to enable. So the other aspect of COVID-TB synergy is at the molecular level. Now, we are all aware the overuse of steroids was done during COVID treatment, and COVID per se would actually knock off the immune response. So it has been clearly shown that the cytokines, etc., showed that there was it was a knock-off effect on the immune system. So what we have noticed, and one or two publications are out and some are under publication, that there was an increased number of TB patients. So there was reactivation TB being reported, higher mortality in patients who had COVID and they got TB. We found that a larger number were dying. So there are individual reports and several such reports which actually are able to show that COVID did lead to a worsening of the TB situation. And by and by, we may learn that there's a increase in the MDR TB being reported. Of course, during the waves of COVID, the reporting of TB by the program was much less because the patients never reached. But the program has been working very hard to make up for the lost contact with the TB patients, and they're able to, their efforts have been fruitful. So though the effort is there, but COVID did give us a bad time.
Okay, ma'am. Thank you so much, ma'am. And I thank all the panelists for taking your valuable time and providing us insights over this topic. And thank you all for joining the webinar.
Thank you. Bye-bye. Bye.
Thank you very much.